Alright, let's talk about excitation, contraction, coupling now.
Excitation, contraction, coupling in GI smooth muscle is how an electrical signal triggers contraction for motility. When a nerve or pacemaker cell, like the interstitial cells of Cajal, sends an electrical impulse, it opens calcium channels in smooth muscle cells, letting calcium enter or release from internal stores, which binds to proteins to spark muscle contraction, driving peristalsis or segmentation. Want to dive into how this links to the enteric nervous system or explore issues when it goes wrong?
Well, I want to understand voltage-dependent calcium channels, G-protein coupled receptors, and how they're connected to all of this clown fest. Calmodulin, myosin light chain kinase, high phosphorylates myosin light chains, and then help myosin heads pull actin filaments along, contracting the muscle cells. Explain all of that to me. Please mention antimuscarinics like atropine and D2 antagonists and prokinetic drugs like in your answer.
All right, let's unpack excitation contraction coupling in GI smooth muscle and tie in all the players you mentioned: voltage dependent calcium channels, G protein coupled receptors, calmodulin, myosin light chain kinase, myosin actin interactions, and drugs like atropine, D2 antagonists, and prokinetics in a clear, concise way, keeping it tied to the clown fest of GI motility. Excitation contraction coupling starts when an electrical signal, like a slow wave from interstitial cells of Cajal or a nerve impulse, hits GI smooth muscle. Voltage-dependent calcium channels, VDCCs, on the muscle cell membrane open, letting calcium flood in from outside, while G-protein coupled receptors, GPCRs, activated by neurotransmitters like acetylcholine from parasympathetic nerves, trigger internal calcium release via signaling pathways like IP3. This calcium spike is the key. It binds to calmodulin, a protein that acts like a switch, activating myosin light chain kinase, MLCK. MLCK phosphorylates myosin light chains, enabling myosin heads to bind and pull actin filaments, causing the muscle to contract, driving peristalsis or segmentation for GI motility. Here's where the drugs come in. Atropine, an anticholinergic, blocks acetylcholine and muscarinic GPCRs, reducing calcium release and slowing motility, often used for spasms but can cause constipation. D2 antagonists, like metoclopramine, block dopamine's inhibitory effect on motility, boosting acetylcholine release and acting as prokinetics to speed up gastric emptying, helpful in gastroparesis. Prokinetic drugs, including metoclopramine or erythromycin, enhance motility by stimulating GPCRs or mimicking motilin, increasing VDCC activity and calcium-driven contractions. The whole process hinges on calcium, orchestrated by VDCCs and GPCRs, with calmodulin and MLCK turning the signal into mechanical work via myosin actin sliding, all fine-tuned by nerves and drugs. Want to dive deeper into how these drugs tweak motility disorders or focus on a specific part like VDCC mechanics?
Oh no, let's continue. I want to ask now about segmentation and about peristalsis.
Segmentation and peristalsis are the two main types of GI motility that keep food moving and processed. Segmentation involves localized, rhythmic contractions of the circular muscle layer in the small intestine, mixing chyme with digestive enzymes for better absorption, while peristalsis uses coordinated waves of circular and longitudinal muscle contractions to propel food forward through the esophagus, stomach, and intestines. Want to dive into how these are controlled by the enteric nervous system or explore disorders like constipation where they go wrong?
Tell me again about segmentation.
By the way, I've posted 2 videos of the outro if you want to check it out. It works perfectly. It works so well. So what,
Can you talk to me about segmentation, please, for mixing? So talk to me about how we have these circular muscle contraction bursts. and relaxation and mechanical mixing. And then for peristalsis, explain to me, how it triggers contraction behind and relaxation in front. I kind of get it. I just don't understand why it relies in the myenteric plexus.
Segmentation is a motility pattern in the small intestine where short, alternating bursts of circular muscle contractions and relaxations, driven by the myenteric plexus, mechanically mix chyme with digestive enzymes, sloshing it back and forth to boost absorption without pushing it far. Peristalsis involves a wave-like contraction of circular muscle behind the food bolus to push it forward, followed by relaxation of muscle ahead, coordinated by the myenteric plexus, which uses sensory neurons to detect stretch and motor neurons to sequence the contraction-relaxation pattern, ensuring directional movement through the GI tract. The myenteric plexus is crucial for both because it acts like the brain of the gut, sensing food's presence and orchestrating the precise muscle firing needed for mixing or propulsion. Want to dive into how these patterns show up in specific GI regions or explore motility issues when the plexus misfires?
I want to ask you a bit about the nervous innervation pathophysiology, for instance. Hirschsprung's disease, rare congenital absence of the myenteric plexus.
Hirschsprung's disease is a rare congenital condition where the myenteric and submucosal plexuses are absent in parts of the colon, usually the distal end, due to failed migration of neural crest cells during fetal development. This lack of enteric nervous innervation stops peristalsis in the affected segment, causing a functional obstruction, leading to constipation, megacolon, or even bowel obstruction in newborns. Want to dive into how this shows up on imaging or explore treatment options?
I want to talk about the process of swallowing. So it's got to do with salivary secretions and muscular contractions. Explain this a bit. Talk to us about the phases: voluntary phase, pharyngeal phase, esophageal phase, and let's talk about the voluntary passage of the bolus into the oropharynx, then the pharyngeal phase, the involuntary passage of the bolus through the pharynx into the esophagus, and lastly the esophageal phase. From the pharynx to the stomach. Or maybe esophagus to the stomach.
Swallowing, or deglutition, is a coordinated process moving food from the mouth to the stomach via salivary secretions and muscular contractions, split into three phases: voluntary, pharyngeal, and esophageal. In the voluntary phase, you consciously form a bolus with chewed food, mixed with saliva to lubricate and start digestion, then use your tongue to push it into the oropharynx, kicking things off. The pharyngeal phase is involuntary, triggered when the bolus hits the oropharynx. Sensory nerves signal the brain to close the nasopharynx, lift the larynx, and contract pharyngeal muscles, swiftly propelling the bolus through the pharynx into the esophagus in under a second, preventing choking or aspiration. The esophageal phase, also involuntary, moves the bolus from the esophagus to the stomach via peristalsis, smooth muscle contractions driven by the myenteric plexus, taking 5-8 seconds, with the lower esophageal sphincter relaxing to let the bolus pass into the stomach. Want to dive into how these phases can go wrong, like in dysphagia, or focus on specific nervous control mechanisms?
So in the pharyngeal phase, the impulses travel to the medulla oblongata and pons in the central nervous system. And efferent signals then drive muscular contractions. Why specifically the medulla oblongata and pons? And let's talk about the soft palate and uvula, how they block the nasopharynx. Let's talk about the epiglottis, how it closes off the larynx. Let's talk about the relaxation of the upper esophageal sphincter. let's talk about the pharyngeal muscle contractions. But
in the pharyngeal phase of swallowing, sensory nerves detect the bolus in the oropharynx and send impulses to the medulla oblongata and pons in the brainstem, which coordinate rapid muscular contracts to propel the bolus into the esophagus. The soft palate and uvula elevate to block the nasopharynx, preventing food from entering the nose. The epiglottis folds down to close off the larynx, protecting the airway, and the upper esophageal sphincter relaxes to allow the bolus to pass into the esophagus, all orchestrated by the medulla and pons in under a second. Want to dive into how these structures malfunction in disorders like aspiration, or focus on the esophageal phase next?
well, I want to understand in regard to the esophageal phase why we have relaxation of the esophageal sphincter and why I guess it's for the food to pass, and then we have coordinated activity of the myenteric plexus and the vagal nerve innervation. Why specifically vagal nerve and myenteric plexus?
In the esophageal phase, the lower esophageal sphincter, LES, relaxes to let the food bolus pass into the stomach, triggered by the vagus nerve, which signals nitric oxide and VIP release to relax the sphincter muscle. The myenteric plexus coordinates peristalsis, sequential contractions of esophageal smooth muscle, while the vagus nerve, part of the parasympathetic system, fine-tunes this motility by relaying signals from the brainstem to ensure smooth, timely movement from esophagus to stomach. Want to explore what happens if the LES or vagus nerve malfunctions, like in GERD?
I want to ask you about achalasia and gastroesophageal reflux now. Tell me about treatment, like physical manipulation or surgical cutting of the LES for achalasia. Explain us the etymology of achalasia too. And tell me about gastric acid secretion inhibitors for treatment for reflux. And also, I want you to tell me about lower esophageal sphincter failing to relax and how this is basically achalasia and tone loss for good reflux.
All right, let's dive into achalasia and gastroesophageal reflux disease, GERD, focusing on treatments, technology, and the lower esophageal sphincter LES issues, keeping it concise as you like. Achalasia is a motility disorder where the LES fails to relax properly due to loss of inhibitory neurons, like those releasing nitric oxide in the myenteric plexus, causing high LES tone and poor esophageal peristalsis. This traps food in the esophagus, leading to dysphagia, regurgitation, and chest pain. Treatments for achalasia often target the LES to reduce its pressure. Physical manipulation, like pneumatic dilation, uses a balloon to forcefully stretch and tear LES muscle fibers, improving relaxation, with about 60% of patients seeing better swallowing, though there's a small perforation risk needing surgical repair. Surgical cutting, such as Heller myotomy, laparoscopically cuts the LES muscle to lower its tone, often paired with a partial fundoplication, a stomach wrap, to prevent post-treatment acid reflux, offering long-term relief in most cases. A newer technique, per-oral endoscopic myotomy, POM, cuts the LES internally via an endoscope, providing similar relief with less invasion, but potentially higher reflux risk since it skips fundoplication. With achalasia, POM might prevent reflux, with less invasion and potentially higher reflux risk since it's per endoscopically invasive.
Okay, let's talk about motor functions of the stomach now. Tell me about the fundus on the upper body of the stomach, how they relax to accommodate larger volumes. Talk to me about how vagal reflex controls this smooth muscle relaxation. And I want to talk a bit about mixing, if you could explain to us propulsion, retropulsion, and cyclical propulsion and retropulsion, how it derives chyme production.
The stomach's motor functions manage food storage, mixing, and propulsion, with distinct roles for its regions and neural control. The fundus and upper body, with thinner smooth muscle, relax to accommodate larger volumes, a process called receptive relaxation, allowing the stomach to stretch without much pressure increase, triggered by the vagal reflex, where vagal sensory nerves detect food entry and signal the brainstem to release inhibitory neurotransmitters, like nitric oxide, via vagal motor neurons, relaxing smooth muscle. Mixing occurs mainly in the antrum and lower body, where stronger muscularis externa drives propulsion, pushing chyme toward the pylorus, retropulsion, forcing chyme back for further grinding, and cyclical propulsion retropulsion, a repetitive pattern that pulverizes food into chyme, a semi-liquid mix, aided by antral contractions and partial pyloric opening. This cyclical action, controlled by the myenteric plexus and vagus nerve, ensures thorough mixing with gastric secretions before chyme trickles into the duodenum. Want to dive into how this goes wrong in disorders like gastroparesis, or explore small intestine motility next?
I want to ask about the regulation of gastric emptying. So, what are these powerful peristaltic contractions we have that force chyme through the pyloric sphincter? And explain to me duodenal enterogastric reflexes of the we have endocrine system and enteric nervous system. Tell me how cholecystokinin mixes with all of this, and talk to me about a pyloric sphincter contraction and decrease of pyloric antrum contraction and how they relate to gastric emptying.
All right, let's dive into the regulation of gastric emptying, keeping it tight and clear. Powerful peristaltic contractions in the stomach's antrum, driven by the myenteric plexus and vagal nerve, forcefully push chyme toward the pyloric sphincter, squeezing it through in small spurts when the sphincter relaxes, typically emptying 2 to 4 millimeters at a time. The duodenal enterogastric reflex, involving both the enteric nervous system and endocrine system, slows gastric emptying when the duodenum senses high fat, acid, or osmolarity. It triggers enteric neurons to tighten the pyloric sphincter and reduce antral contractions, while hormones like CCK and secretin, released by duodenal endocrine cells, further inhibit gastric motility to prevent overwhelming the small intestine. Chyme mixes with digestive enzymes and bile in the duodenum, aided by segmentation, but the reflex ensures this happens gradually by controlling pyloric sphincter contraction, closing it more, and decreasing antral peristalsis.
Okay.
Pacing.
Now, I want you to talk to me about dumping syndrome, gastroparesis, and yeah.
Dumping syndrome and gastroparesis are motility disorders affecting gastric emptying. Dumping syndrome occurs when the stomach empties chyme too quickly into the small intestine, often after surgeries like gastric bypass, causing rapid fluid shifts and symptoms like nausea, diarrhea, and sweating. It's managed with dietary changes or drugs like octreotide. Gastroparesis is delayed gastric emptying due to impaired antral peristalsis or pyloric function, often from vagal nerve damage or diabetes, leading to bloating, vomiting, and fullness, treated with prokinetics like metoclopramide or dietary adjustments. Want to dive into specific treatments or how these show up on imaging?
Can we talk about motility in the small intestine? So we have nutrient absorption. We also have plicae circularis, mucosal villi, microvilli.
All right, let's jump into small intestine motility and its role in nutrient absorption, tying in plicae circularis, mucosal villi, and microvilli. Small intestine motility, driven by segmentation and peristalsis, mixes chyme with digestive enzymes and pushes it along for nutrient absorption. Segmentation, rhythmic contractions of circular muscle, churns chyme to enhance mixing, while peristalsis, wave-like contractions, slowly propels it toward the large intestine, both coordinated by the myenteric plexus and influenced by hormones like CCK. Plicae circularis, permanent folds in the submucosa, increase surface area and slow chyme movement, giving more time for absorption. Mucosal villi, finger-like projections, and microvilli, tiny brush border extensions on epithelial cells, further amplify the surface area by about 600 times total, allowing efficient nutrient absorption into blood or lymph capillaries within the villi. This setup ensures nutrients like sugars, amino acids, and fats are maximally absorbed as motility keeps chyme in contact with the absorptive surface. Want to focus on how motility affects absorption efficiency or explore disorders like ileus?
Why don't we talk about gastric distension, how it promotes peristalsis in the duodenum, and also the migrating motor complex? And then let's talk about the final stages of digestion in the large intestine. Let's talk about water absorption and commensal microbiome, formation of B and K vitamins.
All right, let's tackle gastric distension, the migrating motor complex, MMC, and the large intestine's role in digestion, keeping it concise. Gastric distension, when the stomach stretches from food, triggers the vagal-vagal reflex and local enteric signals, promoting peristalsis in the duodenum by relaxing the pyloric sphincter and boosting duodenal contractions to receive chyme. The migrating motor complex is a cyclical motility pattern during fasting, driven by motilin and the myenteric plexus, sweeping residual chyme, bacteria, and debris from the stomach through the small intestine in phases most intense in phases 3 to prevent bacterial overgrowth and prepare for the next meal. In the large intestine, digestion wraps up with water absorption, where the mucosa reabsorbs about 80 to 90% of water from chyme, solidifying it into feces.
Okay. And can we talk a bit about hostral churning? What's that? Hostral bulges and the defecation reflex?
All right, let's dive into hostral churning, hostral bulges, and the defecation reflex in the large intestine, keeping it short and clear. Hostral churning is the mixing motion in the colon, where hostra, pouch-like segments formed by Taenia coli, contract and relax, driven by the myenteric plexus to knead chyme, enhance water absorption, and form feces. Hostral bulges refer to these pouches filling and distending as chyme moves in, with slow contractions, hostral shuttling, shifting contents back and forth. Mass peristalsis, a stronger wave every few hours, pushes feces toward the rectum. The defecation reflex kicks in when rectal distension from feces triggers enteric and parasympathetic signals via sacral nerves, relaxing the internal anal sphincter and prompting voluntary relaxation of the external sphincter, coordinated by the pelvic floor, to expel feces.
Okay. Thank you so much for watching, guys. I hope you enjoyed this episode. Bye-bye.